Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 2. This is lecture number 39. The topic is gene pools and allele frequencies. Discussing agenda for the topic is gene pools and allele frequencies and the calculation of allele frequencies. Lecture outcome. After watching and listening this lecture, students will know about gene pools and calculate allele frequencies. Introduction. Gene pools and allele frequencies. A population is a group of individuals of the same species that live in the same area and interbreed, producing fertile offsprings. Now here we define a population, so a group of individuals will only qualify this definition if they fulfill those conditions. They must belong to the same species, they must be living in the same area, they must have the capability of interbreeding and producing fertile offsprings. Different populations of a species may be isolated geographically from one another, exchanging genetic material only rarely. It also happens that at times the members of a population gets separated from each other and they rarely get opportunities of mating with each other, and if these two populations are exposed to different environmental conditions, then with the passage of time they will accumulate differences and eventually they will evolve into two different species. Such isolation is common for species that live on widely separated islands or in different lakes. Here is an example where the two populations are geographically isolated from each other. These two caribou populations in the Yukon are not totally isolated. They sometimes share the same area. Still, members of either population are most likely to breed within their own population. But not all populations are isolated. Still, members of a population typically breed with one another and thus on average are more closely related to each other than to members of other populations. So obviously breeding is something where the sexual selection is involved, where the assortative mating is involved, where the selection of mates is involved. So the individuals tends to mate with others that are closely related to each other, either genetically or phenotypically. So more often than not, breeding occurs within the members of the same population living in the same area. A population's genetic makeup can be characterized by describing its gene pool. Gene pool consists of all copies of every type of allele at every locus in all members of the population. Now, suppose there exists a human population in a specific area, and that population consists of 100 individuals. These 100 individuals, humans have 25,000 genes. So in that particular population, the members of that populations have different alleles for the particular loci. Some of them have black hair, some have gray hair, some have white hairs, some have black eye color, some have blue, some have green eye color, some have white skin, some have dark skin. So there are variations in their phenotype. So what makes up a gene pool? Gene pool is the collection of all copies of every type of allele at every locus in all members of the population. So in that population, if you want to develop a gene pool, you will have to consider the gene for every locus, then the different alleles for every locus in that population. If only one allele exists for a particular locus in a population, that allele is said to be fixed in the gene pool, and all individuals are homozygous for that allele. So suppose in our imaginary population, if all individuals have one particular phenotype, one particular trait is common, and it is controlled by a single gene, then we can say that the allele responsible for that gene is fixed in that population. And usually a fixed allele is one which is present in every member of a population. So when an allele becomes the part of the genetic composition of every member of a population, we say that this allele has got fixed in that population. And when the alleles or a gene exist in variants, there are different versions of a particular gene, different versions exist for a particular locus, then we use the terminologies or homozygous or heterozygous when two identical copies are present, homozygous when two dissimilar or non-identical copies are present, then heterozygous. Here is shown an example. Imagine a population of 500 wild plants, 500 wildflower plants with two alleles. So there are 500 wildflower plants, and this flower color is determined by a single gene, and the gene exists in pair form. So there are two alleles for the flower color, and these two alleles are C R and CW. This determines red color, this one determines the white color for a locus that codes for flower pigment. These alleles show incomplete dominance. Now, you know incomplete dominance where neither of the allele is dominant over the other, and both of the alleles are expressed to varying degrees. It is a cross, which is the exception to typical Mendelism. Thus, each genotype has a distinct phenotype. Plant homozygous for this particular allele produces red pigment, means red flowers. Plant homozygous for this particular allele produces no pigment, means white flowers. And plant heterozygous produces some red pigment and have pink flowers. So means pink is a combination of both red and white. You can see here plant homozygous for these alleles red, homozygous for these alleles white, and heterozygous produces pink, which is a combination of red and white. Each allele has a frequency proportion in the population. So now we will move on to the calculation of the allelic frequency in that population. So how to calculate gene frequency and allele frequency? So suppose our population has 320 plants with red flowers. There are 320 plants with red flowers, 160 plants with pink flowers, and 20 plants with white flowers. Remember, these are the number of plants, not the number of alleles. So when you sum up this number, it is equal to 500. So we have a total of 500 plants in that population, and these 500 plants have that gene for determining the flower color, and because the gene exists in pair form, so we will multiply this 500 with 2 and we will get 1000. So there are 1000 copies of the gene, or 1000 copies of the allele determining the flower color. Now we will calculate the allelic frequency. The frequency of the allele that determines the red color accounts for 800 of these copies. Now from where this 800 figure came, so it is very simple. Here is here are 320 plants with red flowers, means both the alleles are CR. So 320 multiplied by 2, This is 320 into 2, and we will get 640. And here 160 with pink flowers. Now remember, you can go back to the previous slide and check 160 with pink flowers. Pink flower has one allele CR and the other allele CW. So we will add 160 with it, and that makes 800. So this is the frequency of this particular allele in that population. Here, now calculating the frequency of CR allele. Now what is the formula for calculating the frequency of a particular allele? The formula is the number of copies of a particular allele in a population. Number of copies of a particular allele in a population. So number of copies of that allele is 800. Here it is calculated, divided by number of all copies, number of all alleles for that particular trait in that population, which is 1000. So we will divide it by 1000 and the result is 0.8. And if you want this value in percentage, then multiply it with 100, you will get 80%. And similarly, you can calculate the allele frequency for CW also. For CW, you can also calculate the allele frequency. These 160 pink flowers means 160 alleles are CW. 160 alleles are CW. And here the 20 white flowers, so 20 white flowers, each flower has 2 copies of CW allele, so it will make 40. So 160 plus 40, it makes 200. So we have 200 copies of CW allele in that population. Now how to calculate the frequency of CW allele in that population? The formula is very simple. Formula is number of a particular allele in a population, means the number of that particular allele, the frequency of whom you are going to calculate. So we are going to calculate the frequency of this particular allele. We will write it here. It has 200 copies divided by the number of all the alleles for that particular trait, which is 1000. And here you will get 0.2. And if you want to get this value in percent, multiply it with 100, you will get 20%. Now when studying a locus with two alleles, the convention is to use P to represent the frequency of one allele and Q to represent the frequency of the other allele. So here in the population which is under our discussion, two alleles are there. One is CR, the other is CW. So we will use this particular symbol to represent this allele and this particular symbol to represent this allele. Thus P, the frequency of this allele in the gene pool of this population, is P is equal to 0.8, just calculated on the previous slide. And because there are only two alleles for this gene, the frequency of the other allele represented by Q must be 0.2. So the other is either you can calculate it in the formula, or you simply subtract it from 1, because in the coming lecture we will study the Hardy-Weinberg equation, which says that the P plus Q will always be equal to 1. So if you know the allele frequency of one allele one allele, then you can calculate the frequency of the other. Here the allele frequency of one is known, so the other is automatically calculated. For loci that have more than two alleles, the sum of allele frequency must still be 100. Sometimes there are multiple alleles where more than two alleles may be present at a particular locus. So in that case you can extend that equation to P plus Q plus R or some other symbol, and still its frequency will be equal to 1. You can put another allele here with a different sign, and then assign a specific phenotype to it and the number of flowers to that phenotype, and then the same formula can be used. The number of copies of that particular allele and the total number of copies in that population. But for now we will not extend this equation to 3 alleles. We will confine with only 2. Next we will see how allele and genotype frequencies can be used to test whether evolution is occurring in a population. So now genotype frequency is very simple. Here from that equation you can also calculate the genotype frequency. The genotype frequency is, suppose we are calculating the genotype frequency for red flowers. Now genotype frequency is the number of individuals of a particular genotype in a population divided by number of individuals of all the genotypes under consideration. So here these three genotypes are present in that population: C, R, C, R, C, R, C, W, C, W, C, W. So here we want to calculate the genotype frequency for red flowers. Now how many red flowers are there? 320. So you write 320 here. All the genotypes which are 500, you write 500 here, and then sum it up. Similarly, if you want to calculate the genotype frequency for pink flowers, how many pink flowers are there? 160. Divide it by the total number of genotypes, which is 500. And similarly for white flowers, how many white flowers are there? 20. Divide it by 500, and then sum it up. And remember one thing, that this and this X plus Y plus Z, this will be equal to 1. This will also be equal to 1. You can calculate it. And so in the coming lecture we will discuss the Hardy-Weinberg theorem. Enough for now. Thank you.